In the last lecture, Katie talked about how diet is a potent manipulator of our intestinal microbiome. Today, we're going to continue our discussion of the microbiome and the food we eat, this time with respect to weight gain and obesity. The first studies to link the microbiome as a factor in obesity involved leptin-deficient mice. Leptin-deficient mice, like the one pictured on the left, cannot produce the leptin molecule that conveys satiation, and thus these mice eat excessively and eventually become obese. Analyzing the microbial communities present in the feces revealed that obese mice had broad phylum-level changes in their microbial communities. Namely, an increased representation of firmicutes and a decreased representation of Bacteroidetes. But what were these microbes doing that was leading to obesity? Let's consider for a moment a simplified model of what leads to weight gain. If the amount of energy we burn off through daily activity equals the amount of energy we consume through our diet, then our body weight is maintained. If the amount of energy burned is much greater than the energy we consume, we lose body weight. Conversely, if the energy consumed is much greater than the energy we burn through activity, we gain body weight. So how are our microbes contributing? Well, researchers began comparing how much energy was extracted from food by the microbes present in a lean and obese gut community. One of the most beneficial aspects of our gut microbiome is our microbes' ability to digest molecules in our food that our own human bodies cannot. Microbes contain genes that produce enzymes to digest these components, such as complex polysaccharides, whereas humans do not possess those genes in our own genome. From an evolutionary perspective, our microbes allowed us to extract the maximum amount of energy possible from the food we consumed, a very helpful feature to have when food was scarce in a hunter-gatherer society. Researchers found that the microbes in an obese community were able to extract significantly more energy from the same diet compared to the microbes found in a lean community. The obese community was able to accomplish this because their microbial members were enriched for genes related to energy harvesting, especially from carbohydrates and lipids, compared to the microbes from lean mice. In our seesaw analogy, this additional extracted energy increases the energy consumed relative to the energy being burned and helps contribute to weight gain. This feature, our microbes' ability to extract energy from our diets, was helpful to our much older ancestors, but can contribute to obesity in modern times when high-calorie foods are so abundantly available. The experiments I just described were correlational in nature, meaning that changes in the microbiome were correlated with changes in obesity rather than proving that an alteration in the microbiome results in obesity. So how can we show causation with respect to the microbiome and obesity? Well, scientists use gnotobiotic mice. You may recall from the first week that newborn babies are immediately colonized by the bacteria they are exposed to from birth. Therefore, gnotobiotic mice have been born in and raised in special bacteria-free chambers so that they are not colonized by microbes in the air and on surfaces. Because these mice are germ-free, scientists can colonize them with new microbes and monitor how these newly introduced microbes affect the health and physiology of the mice. In one experiment, the contents from the distal end of the gastrointestinal tract of obese mice were transferred into the guts of germ-free mice. Over a 2-week period, the germ-free mice that received the microbes from the obese mice gained significantly more weight than did the germ-free mice that were colonized with microbes from the lean mice. This increased weight gain occurred even though both sets of germ-free mice were eating the same amount of food. As we talked about previously, it is likely that the microbes from the obese mice were able to extract more energy from their diet compared to the lean mice, resulting in the weight gain. This experiment helped prove a mechanistic link between altering the microbiome and obesity. As we will talk about in detail next week, the human microbiome has also been linked with behavioral changes, and one interesting example has implications for obesity. Our body's immune system has receptors that help it recognize bacteria. One such receptor is called toll-like receptor 5, or TLR5 for short. Scientists have the ability to raise mice that do not have TLR5. These mice are called TLR5 knockout mice. Mice that have not been genetically modified in this case and have TLR5 are called wild-type mice. In one experiment, scientists were assessing how the gut microbiome and factors involved in metabolic syndrome, such as obesity, blood pressure, and insulin resistance, were affected by mice lacking TLR5. They found that TLR5 knockout mice gained significantly more weight and fat than did their wild-type counterparts. As you can see in this dissection, the TLR5 knockout mouse on the right had far more visceral fat located around their organs than did the wild-type mouse, which had TLR5. More interesting, however, was what happened when fecal samples from the wild-type and TLR5 knockout mice were transferred into germ-free mice. The bacteria from the mice lacking TLR5 actually made the mice hungrier and caused them to eat significantly more food than the wild-type bacterial community. This increased energy consumption by germ-free mice that had received the TLR5 knockout community resulted in increased weight gain. It is interesting to think that your gut microbiome might actually be encouraging you to eat more. Another aspect that has been associated with obesity in humans is the genetic richness of our microbiome. One study found that lean individuals generally harbored microbes that had a greater number of genes compared to the microbes from obese individuals. You can see in this graph that most lean individuals are found to the right of the black line, while proportionately more obese individuals have microbes with lower gene counts and are found to the left of the black line. By analyzing the genes associated with lean and obese individuals, the researchers were able to discover several factors associated with a lean microbiome. Specifically, lean microbial communities had a decrease in the number of butyrate-producing bacteria, an increased ability to degrade the mucin lining the inside of the gastrointestinal tract, reduced hydrogen and methane production, increased Campylobacter shigella abundances, and an increased potential to manage oxidative stress. These more mechanistic insights into the functional abilities of our gut microbiome will hopefully allow researchers to manipulate our microbial friends to aid in weight loss.